Welcome back to another embryology video. In the previous video, we discussed the formation of the neural tube, the spinal cord, and the somites. In this video, we will discuss the brain. Let's take a look at the cranial end of the neural tube. Here, the neural tube becomes pinched to form three primary brain vesicles. They are called the forebrain, or prosencephalon, the midbrain, or mesencephalon, and the hindbrain, or rhombencephalon. The wall of the vesicles will become the tissue of the brain, and the canal inside the neural tube will become the ventricles of the brain and the spinal canal. During the 5th week, the forebrain and hindbrain both subdivide to form the 5 secondary brain vesicles. The forebrain divides to form the telencephalon, which will become the cerebral cortices, and the diencephalon, which will become much of the basal ganglia and the hypothalamus. The midbrain becomes some midbrain structures, such as the substantia nigra. The hindbrain becomes the metencephalon, giving rise to the pons and cerebellum, and the myencephalon, which becomes the medulla oblongata. I appreciate that's a lot to remember. I find it helpful to think about what the lumen of each vesicle will become. The lumen of the telencephalon will become the lateral ventricles. The lumen of the diencephalon will become the third ventricle. The lumen of the midbrain will become the cerebral aqueduct. The lumen of the metencephalon will become the upper part of the fourth ventricle, and the lumen of the myencephalon will become the lower part of the fourth ventricle. As development progresses, the surface of the cortex becomes folded to produce gyri and sulci, with the lateral sulcus becoming apparent by about 26 weeks. The cerebellum becomes distinct at 30 weeks. I'd like to briefly mention two conditions related to the development of the brain. The first is cerebral palsy, which is an umbrella term for a wide variety of childhood movement and postural disorders. The condition presents with spasticity and contracture, ranging in severity from only one side of the body affected to all limbs affected with severe scoliosis. Prevalence is stable, with around 2 to 3.5 cases per 1,000 live births. A major risk factor is low birth weight, although about 10% of cases are post-neonatal, generally due to central nervous system infections. Neonatal asphyxia is also thought to be a key cause of cerebral palsy, suggested by the increased risk for monochorionic twins. Maternal infections such as cytomegalovirus and rubella are also major risk factors. The second condition is fetal alcohol spectrum disorders. These are a range of neurodevelopmental disorders related to prenatal alcohol exposure. Fetal alcohol syndrome is the most well-known part of the spectrum. Alcohol is harmful to the fetus via direct apoptotic damage to developing neurons and cells. This can interfere with neural folding, migration, and connectivity, which all occur in the 3rd trimester, leading to cognitive deficits and developmental delay. FAS is associated with the classic features of lack of philtrum. The philtrum is the bit from your nose to your upper lip, a thin upper lip, and thin palpebral fissures, which is the distance from one side of the eye to the other. So what is a safe level of alcohol during pregnancy? Well, high intakes lead to clear neurological and physical damage. Lower intakes are more complicated. Individuals differ, and while for the majority low intakes are not considered harmful, this does not seem to apply to all children. In the end, there is unfortunately no clear safe level, and abstinence for now is the only way to avoid any risk to the unborn child. Thank you for watching, and we'll come back to further developmental disorders in the next video.
Subscribe to Soton Brain Hub for more videos to help explain the mysteries of the brain.